Okay. So in the nucleus, in the animal cell, there is the nucleus. Nucleus contains the nuclear membrane, okay, and it's contain the liquid portion of the nucleus is the nucleoplasm. Okay, the liquid portion of the like that cell is known as cytoplasm. So the liquid portion of the nucleus, inside the nucleus, because nucleus is surrounded by a membrane, so nucleoplasm. Okay, now one important thing related to the nuclear membrane is that nuclear membrane is this is the nuclear membrane, and it is porous. Okay, and wherever the pores are present, okay, if we talk about, like, if this is the nuclear membrane, so the pores are here, so it's a porous membrane. So from porous membrane, there are extending the endoplasmic reticulum. Okay, they are fitted on the pores, so it's like a tunnel. Like if this is the nuclear membrane, it's whole, it is these pores. So now endoplasmic reticulums are opening from it like that. So so that whatever is being synthesized in the nucleus, in the genes or in the genetics, will then transfer into ER. Okay, endoplasmic reticulum, whether the smooth or the rough endoplasmic reticulum. Okay, so all the eukaryotic organisms are covered by a nuclear membrane. Okay, and we are in this chapter we are discussing the eukaryotic and cells. So it contains the chromosome. So this structure is known as chromosomes. Okay, now what is this chromosomes and what are its part? Okay, this is the sister chromatids. Okay, this one arm is containing one set of genes, and the other arm is containing the other set of genes, but contains the, like, what we say, copy, locus of the same gene, alleles. We will discuss all the terminologies, like what is allele and what is the thing. Like for example, if there is a allele of eye color gene, and there is another allele of eye color, so they will be in the same positions in the two arms. Okay, so now these can come from different things. One can come from father and one can come from mother. But in the human genome, wherever the eye color gene is present, will be there. Okay, one from the maternal side, one from the paternal side. So for example, the father color eye is blue, mother color eye is green. So that person has the two variants of the eye color in it, or him or her. So these two arms are known as sister chromatids. Okay, sister chromatid. This is the one, and this is the one, this is the second, and they both are attached together by a centromere. Okay, it is very important factor while these cell division is taking place. So this centromere plays a very important role in the replication. So you have to know these names: centromere, sister chromatid, and the chromosomes. Human cells contain the 46 number of chromosomes. Like these chromosomes, whatever these are in the tiny nucleus, so they are 46 in number. Okay, and it is specific. These number of chromosomes are specific for the species. Like humans have the 46, chimpanzees have the 48, then Drosophila and different kind of other organisms have the different number of chromosomes. And if these number fluctuate, like if the number are changed or number are changed, so like if we have the 48 chromosomes, so we can become the chimpanzee. Okay, so chimpanzees has two more extra chromosomes than us. That's why they are different. They are other species, we are other species. So these are, and it doesn't depend upon like how the organism is small or the organism is big. The number of organism or the number of chromosomes doesn't depend or doesn't say like if it has the more chromosomes, so the organism is the big. It doesn't reflects that. Okay, so it can be possible, that might be possible a organism is this much big and it may have only one chromosome. Okay, or it can be possible that organism is so small but it has so many uh, chromosomes in it. Okay, so it doesn't depend upon the size of the organisms, the number of the organism, but the chromosomes, like it depends on the complexity of the organism, like how much any organism is complex. Like as I told you while teaching you about the retina, in that chapter we discuss about the rods and cones. Okay, so we as a human, we can detect only few colors. Okay, there is a range of red, blue and green. Okay, so we can just see the colors in this spectrum, in this limited spectrum. But the birds have the different spectra. They have the more genes, more active genes of the different colors, different variants, different cones. So there are different genes that are responsible for the rods and cones. So that's why those animals can like see better in the night. They can see a variety of the colors. They can even detect the levels, depthness, because they have to fly, so they have to like their eyes must be really sharp in order to like uh, take a understanding of the each and everything. Like they can uh, perceive everything in a very detailed way. So the number of chromosomes are really like we have 46 chromosomes. So these 46 chromosomes contain the genes that are necessary for a human. The chimpanzee contain 48 chromosomes. So it means it has the two extra chromosomes that has the genes that are essential for the chimpanzee only, that are not essential for the rest of the organism. So it depends that which organism, which chromosome is containing the which kind of gene. So if we just let's suppose if I have a chromosome and it has the genes of like eye color and like leanness, then it has the color of um, straight hairs. Okay, now if I have the other chromosome and this other chromosome has the different kind of genes, like this gene is regulating the blood pressure. Okay, this gene is responsible for the uh, muscles, good muscles, healthy muscles. This gene is responsible for the what to say, good bones. Okay, let's suppose good bones. So now if I will just put this chromosome, it has the different genes to the chimpanzee. Okay, if I will put this like this chromosome has the genes of good muscles, good bones and the blood pressure, or okay, so if I will put this chromosome in this chimpanzee and now making it 49, so it has now these genes in it. So although it has the 49, now it has the 49 chromosomes, but its cell will express, now its body will express these things. Okay, Now, if I have the another organism with naturally occurring organism that has the 49 chromosomes. For example, suppose it's not true. Let's suppose if a cat has 49 chromosomes. Okay, in actual cat don't have the 49. Let's suppose. So this would be different. Okay, it's not like that chimpanzee would become after receiving one chromosome into the cat. It's not like that. Chimpanzee receives the chromosome of the different set of gene. So its genes will synthesize the different proteins. So it will become something different, some different organism. It can become like, it can have the extra hand on it, or it can become any other kind of organism. Okay, so in the many conditions, in the disease conditions, when the humans have one lost chromosome and one extra chromosomes, so they are the humans, but those are the disease conditions like the Down syndrome. Okay, so they will remain the humans, but they have one extra and one lost copies of the chromosomes. So only the number of gene, the proteins are lost, so that they will be deficient in that protein and they will be efficient. Now if I would like to, like in this chimpanzee, I would add the genes which are responsible for making this a chimpanzee. If I will take those chromosomes and I will add those additional things in the human, so now the human can become a chimpanzee, because those were the distinctive chromosomes and those were only found in the chimpanzee. Those were not found in the human. So there's that distinctiveness I have introduced now in the human. So now the human is now able to synthesize those proteins, and now it can synthesize the protein that are present in the chimpanzees, that are useful for the chimpanzee. So the physical features, physical appearance of a human can now in that sense can become or resemble like a chimpanzee. Okay, so it's all the game of which protein, which gene you are you want to introduce into that particular species. So species wise they vary, and every species contains the chromosomes or the genes that are relevant to their characteristics. Like if I have a fly, if there is a fly, so fly needs or it's a honeybee. So honeybee needs the features that are required for making the honey. Okay, it sucks the nectar of the flower and convert this nectar into honey. So this is the only speciality of a honeybee. Humans cannot do this, but the genes that are responsible for converting this nectar into honey, if those genes I will introduce into human cell, so now human can develop a tendency to synthesize the nectar into the honey. Clear? Or if I will just introduce this gene into a normal fly. Okay, if this is a normal fly and normal house fly, so if I will use this gene of the honeybee and I will introduce this in an ordinary fly, so now this ordinary fly can now be able to synthesize the honey, because it has now the gene of the honey making or honey synthesizing. So you have to like very clear about the chromosomes and the clear about the genes and the chromosomes that are specific for containing the specific kind of genes. So now if we just open this chromosome, okay, it is just packed, and it's just packed same as like a reel of a, like when while we are stitching the clothes, so thread reel. Okay, so this is same like that pile in the way. If you got a chance to open up a thread reel, So thread reel is, you will find two threads like that in that reel. If you just open a thread, so there are two threads that are intertwined with each other, and those are now rotated or wrapped around the cylinder. So if we just open this thread, so there are two threads, and these two threads are combined in like they gave the appearance of a single thread, but they are wrapped around a cylinder of any cardboard. So same way the nature has wrapped the DNA molecule. Okay, DNA molecule, like what is DNA? Deoxyribonucleic acid. Okay, and AGTC. So what is AGTC? Adenine, guanine, thymine, and cytosine. And the DNA molecule in the chapter biological molecule, you may have studied about the DNA molecule, but if not, so it's a, like DNA molecule consists of three things. One is ribose sugar, second is nitrogenous bases, nitrogenous bases are these: adenine, guanine, thymine, cytosine. So these are nitrogenous bases. And the third one is the phosphatine. So how now these three molecules make up a DNA molecule? Ribose is basically a pentose sugar. Like the glucose is a hexose sugar. Hexose means 6, pentose means 5. So while when I draw the structure of glucose, so it's 6 carbon ring, okay? And when we are talking about the pentose sugar, so it's a pentagon, like that. And here in this pentagon, at the very first position, there are the nitrogenous bases. Okay, like for example, if there is adenine, and then at this point there is phosphatine. Okay, so this is the one DNA molecule. Now if we take the same, okay, and now this is the another DNA molecule, and here instead of adenine, there comes the guanine. Okay, and so on, thymine and cytosine. So they all comes like that way. So here at the first position attach the nitrogenous base, and at the fifth position, one two three four, fourth position, there adds a phosphate group. So this becomes a DNA molecule, and they both are, these two molecules are like interacting, these adenine and guanine are interacting with each other with the hydrophobic ends. Okay, but that is of not your concern and not of your syllabus. Hy Hydrogen bonds. Okay, so and so on, these molecules are arranged in a pattern that they forms a helical structure, like that. First of all, the DNA is a complementary molecule. Okay, so this is the one molecule. For example, adenine is here. If we just, and it's a complementary strand. If we just open up this one strand, okay, so it will appear like that. So if there is A, so there will be T. Adenine always binds to thymine. Cytosine always bind to guanine. And adenine and thymines are bound together with the two covalent bonds, and cytosine and guanine binds with the three covalent bonds. Okay, so this bond is very strong and it needs the very high temperature to be broken down. And while the adenine and thymine are like, they have only two bonds, so they can, like, upon receiving the high temperature, they can easily melt. So adenine will bind to the thymine, AGCT, TCA, C, G, A, A, G, T, T. Now you should know the sequences. In MCQs, this can come, they can give you the any sequence, like for example, A, T, C, G, T, T, A, A. Now you have to choose from the option the complementary sequence. So how it would be a complementary sequence? If you know this formula, A will always bind to T, C will always bind to G. So when there is A, you have to write T in the second strand. A will bind to T, then C will bind to G, G will bind to C. Now these T will again A, and then TT. So now this strand is a complementary strand of this parent strand. Okay, so this is the parent strand, or the template. We can also say it template. And this is the complementary strand. So these two strands are bound to each other. And if I'm making this, so obviously there is now a pentagon and a phosphate. Again a pentagon and a phosphate. Again a pentagon and a phosphate. And so on the chain continues. And same like that, in that way, pentagon and a phosphate, pentagon and a phosphate. So and so like that. So now the interactions between the phosphate ions cause the bending of these helices. Okay, the turn that is occurring is due to the repulsive and attractive forces between these ions. When they are close, it means there is the attractive force. When they are bending from a point, so it means there are repulsive forces. So they are arranged, these DNA molecules are arranged in a double helix. And it's like a twisted ladder. Clear? Any question?
No, miss.
And it's like that. Okay, so now what are genes? Okay, if we talk about, or if we want to make our concepts clear, so this one sequence is the gene. Okay, it can be of anything. Okay, now let's go to I will just show you like how the sequences are. Okay, if I want to search any gene, like let's start with the any normal, any protein, like if we search for example amylase. Okay, let's search the amylase gene, that where it is present. So amylase is produced by different organisms, different things. So we want the human amylase. Okay, that okay, different organism amylases are there, but we need the human one. I would just, okay, so now this is the human amylase, and here this is a database in which every information is available. So you can see here. Okay, this is the very advanced thing that I'm like telling you, which we do when we search out any gene. Okay, now here in this picture you can see that these are the organs where the amylase express. Okay, so this is the data related to these samples. They have taken each and every organ of the human and try to find out the secretion of amylase. And we also know that amylase only secrete in the two sites, in the saliva, from the saliva, and from the pancreas. So here it is also showing that human amylase, in human body amylase is secreted only in the salivary glands, because it has to break down the sugar, and in the pancreas, where it again has to break down the sugar, remaining sugar. So you can see here, you can verify from here with the like scientifically. Now this is the chromosome structure, but this is the chromosome. Okay, so now here you can see they are telling the location of the amylase gene. Okay, so amylase, this is the chromosome number 1. Okay, and in the chromosome number 1, p arm 21.1. Okay, so this is the code that you at this level not understand, but they are telling that this is the chromosome number 1. Rest of the things are related to the arms. Okay, like the chromosomes have the arms p and the q arm, so that is denoting the p arm, and 21 is the location of the band, and one is one may be also describing something. So this is the location of the gene. Amylase gene is present where, in which like chromosome. Now if I have to I was looking for the faster sequence. Okay, here if you can see that this is the sequence. Okay, this is the protein sequence. Basically I'm looking for the gene sequence. Okay, so now you can see here DTV, these sequences. Okay, this is in terms of protein. Okay, like from these are the protein codes like amino acids, like alanine, glycine, valine, and there are so on. So these codes are for the uh, amino acids, but same like that the sequences of the genes are also there. So like AGTC, you will find the complete sequence of the AGTC sequence. Yes, like that. Okay, can you see this screen? Can you see this screen?
Yes, miss.
So you can see that how many C, T, T, C, 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 and so on. And it's a very long sequence, like it is ending here. So these are the codes that are only responsible, this long code is only responsible for the amylase gene. Okay, amylase protein. So when these genes are activated, so they synthesize the amylase protein, and in the humans it is on the chromosome number 1. So everything now, if we talk about, like, we have 46 chromosomes, so it means we have so many genes. Our like one chromosome is very long, 2 meter long. One chromosome, the DNA molecule, double helix molecule, is wrapped around a chromosome is 2 meter. Okay, 2 meter long. So it's a very huge thing, very huge. And if every chromosome, like this is gene 1, this is gene 2. So it's a very long thing. And there are so many genes that are regulating and performing their tasks. So, okay. Now, when we have the, now you should know this sequence that is known as central dogma. DNA, DNA when opens, okay, when it has to make any protein, when it opens, so it needs a template to be bound with it. So first DNA needs a RNA molecule, and first DNA is converted into RNA, and then RNA is then converted into protein. Okay, so this sequence is known as central dogma of biology. So when any genes wants to be like synthesized, or if any gene, so first the DNA molecule that is in the double helix opens up, and then, for example, here is the gene that is looking for the synthesis. So A, C, G, T, T, T. Now I need this protein to be synthesized. So how it will be synthesized? This protein, in our cytoplasm there must be the ribosome, or the, what we say, RNA molecule. Okay, RNA is a molecule that is it contains A, G, instead of T there is uracil. Okay, and then C. So this molecule contains this sequence, and this is act as a template. So when this molecule comes, for example, the complementary RNA molecule of this gene would be A, uh, C, no, complementary would be T, G, C, and instead of all T, there would be U. Okay, so now this whole RNA molecule will bind to this side. This will bind to this will bind to this T, this U will bind to this T, this. Okay, so now this RNA template has now bound with the DNA molecule. And now when it binds, it starts to synthesize or manufacture the protein. So now this gene, which is, for example, responsible for the color of eye, and that is blue color. So now this, when the RNA, relevant RNA molecule is bind to it, it will start to synthesize the blue color eye gene protein, because that protein, because when we are seeing the blue color in the iris, okay, iris is forming the color. So this blue color, this part is basically in real when you touch it, when you hold it, when you, so it is a protein in actual. So our body, when our genes are synthesizing this blue protein, so this code, when RNA will bind to this gene, so it will start to synthesize the protein. And when this protein start to synthesize, so, and so many protein, like until or unless you will achieve the desired concentration, the proteins will continue to be synthesized. Same as like that, my computer is a contains us, like I'm opening a Word document and I have typed a document, and now I want this document in the hard copy. So for that, I need those instructions to be given to the printer. How I am going to convert this soft in the hard copy? So I will give the commands, instructions to the printer. So I will go to the print option and then I will select. So for that, before printing, I need some template to be print out. Okay, I need something to be print out. So that template is the RNA, and that will first bind to the DNA. And when the command is given, when they are attached together, they will generate the synthesis of protein. So when the print command is selected, so the printer start to make the copies of that document. I need the 100 copies, it will print the 100. I need the 200 copies, it will make the 200. So how many amount of the protein I need, my body will synthesize the same protein. So now this protein, this eye color protein will go and accumulate in the iris. Okay, this will go and accumulate or synthesize in the iris. But it's not like that this gene will activate on my hand. Okay, this gene will only activate in the cells of the iris. Clear? So this gene cannot be activated on my hand. So it is silent. So there are two principles when we are talking about the activation and inactivation of any gene. So we use the terminology on and off. So when the gene is, when this DNA molecule is wrapped, so this is unactivated. Okay, this is not synthesizing any protein. If it wrapped around the chromosome, so it is inactivated, it is not synthesizing anything. But when it unwinds and open up like that, okay, so now it's activated. So only that portion will be activated, that portion will be open, which protein needs to be synthesized. Like if I have a cupboard, and a cupboard has so many files, archive of so many things. In our office, I have a cupboard, and this cupboard has so many shelves, and it contains so many files in it. a number of files, record of the previous student, this and this. And if I will not organize it, if I will don't know this is section 1, this is 2, this is 3, this is 4, if I want any document, I will go to the specific part and I will just take the thing from there. So same like that, when any protein needs to be synthesized, only that chromosome unwinds, and only that portion of the gene is unwind and opens up. And when it is open up, now it can synthesize the protein. Rest of the portion is unactivated, rest genes are off. So on my hand, the cell of my hand, the gene of the eye color are inactivated on my hand. Although my hand chromosomes are containing the eye gene, but the eye color gene is inactivated on my hand. It is activated only in the iris tissues, iris cells. So because the protein, when the iris cells will synthesize the protein and the protein is being synthesized, so automatically, if this is the iris cell, okay, and these are the iris tissue. So if these tissues are synthesizing the protein, so obviously there would be large concentration of this protein in these tissues only. Rest of the genes are not expressing in these tissues. So automatically our color of eyes will look blue. Clear? Clear? Any question? So inheritance is the transmission of genetic information from one generation to the another generation. So how this transmission occurs, like if we want, so every cell we have the 46 number of chromosomes, and these 46 number of chromosomes are in our somatic cells. Okay, these are the normal cells, hair cells, hand cells, every cell. But there are the half number of cells, 23 and 23, in our sex cells like egg or sperms. So half set of chromosomes comes from our mother and half set of chromosomes come from our father. Half set of gene. So this half, when mother and father cells combine, so they the new offsprings have a 46 chromosomes. So now whatever the features of the father, if the offspring is carrying, so that is inheritance. Like blue eyes, so offspring also have the blue eye. So it means it inherited this from father. And now if it is having like he is having anything from the mother, for example, let's suppose straight hairs. Mother have straight hair, so the offspring also have the straight hair. So this is inheritance. So now this person has the blue eyes and the straight hairs. Okay, father have the curly hairs, not the straight ones, and mother have the green eyes instead of blue. So now which features are dominating in the offspring? Blue eye and straight hairs. Okay, so these are the features that are dominant in the offspring. Now, which is not dominant in the offspring? So curly hairs are not dominating in the offspring. Curly hairs, although the offspring is also carrying the gene of the curly hairs in him, but they are not dominated in its physical appearance. Okay, so they are known as recessive. And if and here, if in case of mother, so green eye is not a dominant feature of the offspring. So this gene is now not dominant. So now this green eye, although the offspring inherited this color of eye, but it is not expressed in its eyes. So that's why the green color eye is also recessive. So here two terminologies are very important when we are talking about the inheritance, that is dominant and recessive. So you should know like which characteristic is dominant and which characteristic is not dominant. So as here you can see, so this is known as hereditary, and in which in the branch of biology in which we study these things, they are genetics. So for example, this is the cat, it has the blue eyes, and there is another cat, male cat, which has the golden eyes. So now the offspring now expressing the both colors, blue and golden eye. So this is the inheritance. Like it's one eye, in the one eye, the protein of the golden eye are expressing, while in its another eye, the protein of the blue eye are expressing. So both are expressing. So that's why it's expressing the both the colors. So this is the inheritance. So now protein, a chromosome is a thread of DNA made up of a string of genes. Uh, the gene is a length of DNA that codes for a protein. These all things I have already told you. Okay, so this picture is a karyotype. Are you listening? Ali, are you here? Hello? Hello, hello, hello. Okay, so this is the human karyotype, and this is the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and the last pair is the 23. So in the last, we have two kinds of chromosomes that are known as X and Y. Okay, so humans have the karyotype and it has the 23 chromosomes. So this 23rd chromosome is the X and Y. So if a karyotype has a X and Y, so it means it is a male. And if this last pair of chromosome 23 is X and X, so it is reflecting the female. So if we have the half number of chromosomes, like if this is the father and this is the mother, XY, XX. So this is producing two half and two half. This is producing one X and one Y. This is producing X and X. So one offspring like this one chromosome will bind to this chromosome. This one chromosome has the possibility to binding with this chromosome. This chromosome has the possibility of binding with this chromosome, and this chromosome has the possibility of binding with this chromosome. So this will become XX. This X and this Y and X will become XY. This X and this X will form the XX, and this Y and this X will form XY. So now there is the chances of producing offspring. That is, this is the girl, this is the boy, this is the girl, this is the boy. So this feature that is reflecting the genetics, okay? So this is known as genotype, and this which is reflecting the physical appearance, like apparently this is a girl, by physical appearance this is the boy, by physical appearance this is a girl and this is a boy. So this is known as phenotype. Let's take another example. If I have a gene, if this is the mother chromosome and this is the father chromosome, if mother has the blue eye and father has the green eye, and if the mother have brown skin tone and the father has white skin tone, okay, white skin tone. So when the these chromosomes will split into one characteristics. So if we talk about the characters, so this will form two. If this will split in the half, so there would be blue, there would be green, there would be brown, and there would be white. So if I want to cross them or made these characters, so how it will be going to produce the effect? So if this chromosome will be binding with this one, okay, and this chromosome will be binding with this one, this chromosome will bind to this, and this will bind to this. So it will be producing the effect. There will be the blue color and the green color. There will be the skin tone and the eye color. Okay, let's make a checkerboard. Okay, so we have the blue eye color gene resembling with the capital and a small b. Capital B is resembling blue, and a small b is resembling green. Now we have the skin tone. So capital Y and a small y. So capital Y is resembling the white color